A-Level Biology 9700, Paper 4, Variant 2, May, June 2025, question number 3, second video on this paper. So we will start with question number 3. Rice Harissa sativa is an important grain crop. A rice grain is a seed and can have a structure known as an awn which projects from the tip of the grain. 3.1 shows a rice grain with an awn present and a rice grain with no awn present. The development of awns is controlled by two genes, capital A, capital B, small b. So the genes are present on different autosomes. The presence of either dominant allele capital A or dominant allele capital B results in rice plants producing grains with awns. So a cross was carried out between a rice plant that is homozygous dominant for the two genes, double homozygous dominant as a rice plant that is homozygous recessive for the two genes, double homozygous recessive. All F1 offspring plants produce grains with awns. So construct a genetic diagram including a Punnett square to show the cross that produces the F2 generation including phenotypes. So state the ratio of the offspring phenotypes produced. So here they have, that is the with capital capital letters show the characteristic of dominance, whether it is capital A or capital B. Dominance, and small letters, they show the recessive characteristic. So there are two parents, plant 1 or parent plant 1, plant 2. They are the parents, and plant 1 has double homozygous dominance. So all dominant letters, small a, small b, double recessive characteristics with awns, no awns. So if we cross these two, double A, double B, into small a, small b, so the F1, for F1, the gametes formation AB, AB. So there will be AB, capital A, capital B. On the other hand, there will be small a, small a. So F1 will be capital A, capital B, small b. So the to get F2, there will be capital A, capital B, small b into, so there will be the self-fertilization of F2, F1 seeds to get the F2. So here we will make a Punnett square after making the gametes. F1 gametes are capital A, B, A, B, capital A, small b, and then there will be small a, capital B, and small a B will be on both sides, the same. So we will make a Punnett square for them as AB, capital A, will be S. On both sides. So it will be capital A, capital A, capital B, capital B, capital A, capital A, capital B, small b, capital A, small a, capital B, capital B, capital A, small a, capital B, small b. So capital A, capital A, capital B, small b, capital A, capital A, small b, small b, capital A, small a, capital B, small b, capital A, small b, small b. So capital A, small a, capital B, capital B, capital A, small a, capital B, small b, small a, capital B, capital B. So small a, capital B, small b. So capital A, small a, capital B, small b, capital A, small b, small b, small small a, capital B, small b, capital B, small b, and then small a, small b, small b. According to the rules of inheritance, you know that the when there is capital letter, it shows the with ons, ons, with ons. Because you can see capital letter, a single capital letter shows the dominance. With ons, with ons. It is a double hybrid cross. Double hybrid cross, you know, 16, 9:3 ratio. With ons, with ons. With ons, with, because a single letter is all in all. Now you can see this one. All small letters are there. It means it will be with no ons. So if we see that out of 16, there is only one, which is without on. So for this, we can say that there is only one out of 16. The ratio can be 15:1. So in this case, 15 with ons and 1 no on. So it was all about this Puneet square. Now moving towards the next part of this question, that is deduce the type of inheritance shown by the ratio of the offsprings present stated in 3a1. So in this case, you can see the F2 generation ratio was 15:1. So you can say uh, this is the characteristic of dihybrid inheritance. Dihybrid inheritance. Dihybrid inheritance, and you know the epistasis is there. Dominant epistasis. And what is epistasis? Epistasis is the presence of a dominant allele at either locus is sufficient to produce the dominant phenotype. That is, only one can produce uh, one can produce a complete characteristic. So here you can see epistasis. In simple words, I would like to say that epistasis is a general phenomena where the effect of one gene is masked or modified by one or more other genes. So this is a type of gene interaction where the presence or absence of an allele at one gene locus depends on the allele present at the other locus. So the interaction influence physical traits and can complicate inheritance patterns. So only one can. So this is the type of epistasis, which is dominant epistasis. And this is a type of dihybrid cross, dihybrid inheritance or dihybrid cross. Now the is a common for wild rice plants to have grains with awns present. Suggest a selective advantage to wild rice of having awns present on their grains. So the awns are, long needle-like projections on the grain. So that is the way of protection. They provide protection. They provide protection being needle-like. Being needle-like. And second point can be that is they can make the grain difficult or less palatable for animals. And third can be, we can write that help space out grains for better air circulation. Now, the next is One of the changes that occurred during the domestication of wild rice to cultivated rice was the loss of the awns from rice grains. Farmers found the long awns made storing and processing rice grains more difficult. It was also observed that rice plants that have grains with no awns have an increased grain yield. Explain the principles used by farmers to produce the rice grains with no awns. So the question's next part. So the long ons made storing and processing rice grains so more difficult. So the it was with no ons have an increased uh, and what happens the loss of ions during example of artificial selection. You can see that this is the example of artificial selection. Artificial selection where humans, where farmers selected traits beneficial beneficial for survival in the wild. The loss of horns provided a selective advantage. Not the plants. So this is the advantage to the farmers, not to the plants. So uh, unless varieties were selected, varieties were selected because they were they were easier easier to harvest, transport, and thresh. So they select them and store in bulk. So they adopted the method of artificial selection, which take many generations to get the required product. So if the all-less plants had an associated increase, farmers would have selected this species to increase productivity. Because the resources are diverted from protective to reproductive. That is from ons to no ons. So it was the question number 3 is part C. Now the second part of question is the normal allele for the gene AN1 codes for a transcription factor that has a role in on development and in the number of grains of rice produced. When the transcription factor is present, there is an increase in the expression of genes involved in on development. Positive regulation decrease in the expression of genes involved in the number of grains produced. Negative regulation, so suggest and explain how changes at the AN1 locus can cause rice plants to have grains with no ons and increased grain height. So we have to write the answer over here, and the according to this question, which asks you to link a change in a regulatory gene, which is AN-1, to the desired domesticated replication traits, no ons and increased grain yield, based on the provided information about the gene's function. So we have to suggest and explain the change at the AN1 locus. So the goal is achieve two outcomes: no ons or increased gain yield. So first of all, the the normal allele of AN1 codes for transcription factor, which when present when present. So it will affect the gene expression, which will be on resulting effect on gene expression. So a mutation occurred at the AN1 that result are non non-functional transcription factor. Which causes the AN1 gene to produce no protein or a protein which cannot bind to DNA or function as an active transcription factor. Since the transcription factor is absent, there is no increase in the expression of genes involved in on development. So this is the positive regulation. Now the negative regulation is that since the transcriptional factor since the transcriptional factor is absent. Absent. It can no longer. Decrease the expression of genes. Expression of genes involved in producing the number of grains. The inhibitory effect is removed. Leading to a higher expression of genes. Genes which give high yield. So it means it will act as both way, that is the positive regulation and the negative regulation as well. So it was all about the question number 3. Please like, share and subscribe my channel to watch more videos on these papers. And thanks for watching.